right, thank you, Steph, and thank you, everyone. I'm glad to see some infectious disease aficionados in here today. As Steph mentioned, my name is Troy Hubbard. I'm a graduate student in the Biological and Biomedical Sciences PhD program at Harvard Medical School. And I work in a research lab that studies bacterial pathogens of... Oh, louder? Do we have a volume option on this?
Just move the mic up.
Ah, okay. How's that? Better? Yeah. Okay, cool. Sorry about that. Yeah, so my lab studies bacterial pathogens of the intestine, and these are bacteria that can colonize your intestine and cause disease. Goodness, okay, we're off to a bumpy start. So today I'm going to be talking about a very specific disease of the intestine called cholera. And I'm going to begin with this discussion of the Broad Street Cholera epidemic, which really kicked off the modern era of cholera research, and it also transformed the way that we think about infectious disease. Next, I'm going to move into a discussion of our current understanding of cholera, including the cause of this disease and how it's transmitted through a population. And finally, I'll finish up with the discussion of cholera in the modern world, focusing specifically on an outbreak of cholera which struck Haiti in 2010. The message that I'd hope to leave you with today is that with many outbreaks of infectious disease, and with outbreaks of cholera in particular, there is an inherent social context. These diseases aren't really random events. They're often precipitated by a set of societal factors, and first and foremost, they're brought about by poverty. So we here as scientists can develop all sorts of effective therapies and even cures for these diseases, but being able to treat a disease doesn't address the underlying set of social factors which can bring about an outbreak of disease to begin with. And I think there's no better illustration of the social context of infectious disease than the fact that we here in the United States don't have to think about cholera. In fact, before I started studying infectious disease, the only exposure I had to cholera was through Oregon Trail, and then also through like the dust jacket on Love in the Time of Cholera. But I've never had cholera, and I'd be willing to bet that almost no one in this room has either. And what's even more of an illustration of this is that even if by some unfortunate set of circumstances, all of us were to contract cholera this afternoon, and this is a hypothetical, this is not a threat, we'd all be fine within a couple of days because we have amazing therapies and basically cures for cholera. That being said, it's pretty shocking that cholera is still a major cause of preventable suffering and death around the world. Each year, between 3 and 5 million people contract cholera, and it's responsible for over 100,000 fatalities. And while this death rate may seem very high for a disease with such effective treatments, it's actually much lower than it has ever been throughout the entire history of humanity. This organism has been with us for a long time, and it actually, before modern treatments, typically killed more than half of the people that it infected. And that was probably the case in 1854 when when cholera struck the Broad Street neighborhood of London. And the reason that we think about this Broad Street cholera epidemic in the modern era is because of the work done by this man here, John Snow. And this is not Jon Snow, like defender of the realm, but he is a defender of the realm in another sense, in that this man was a physician. He was probably best known for his work studying anesthesia, but he actually had a long-running experiment looking at the quality of water that people drank in London and their infectious disease status, whether or not they contracted things like cholera. So at the time, the prevailing model for how someone contracted a disease like cholera was due to this concept called miasma, which is sort of this vague notion of bad air or noxious fumes. It was thought that if you were exposed to this miasma, it smelled bad, so it would make you sick. But John Snow didn't really subscribe to this mindset. He believed that infectious disease was more likely associated with some sort of contagion or an infectious particle that you could contract potentially through water. And so when he learned of the Broad Street cholera epidemic, he began a massive study where he basically went to the Broad Street neighborhood, started knocking on doors and asking people, where did you get your water? How are you feeling today? Where do your neighbors get their water, and are they sick? And so he interviewed hundreds and hundreds of people over the course of this epidemic, and basically took his data and overlaid them on a map of the neighborhood. So what you can see here in these horizontal lines is actually a fatality associated with cholera during the Broad Street epidemic. And each line is placed in the residence where that person lived. And when John Snow looked at this map and then put together the data where these people got their water, he was shocked to find that almost every one of these vertical lines represented a person who drank water from the Broad Street water pump right here on the corner. So he took this data to a board of local authorities and said, look, people drinking from this pump are dying from cholera. We need to stop this. And so the local authorities, kind of skeptical of his results, removed the water pump handle so that no one could drink from that water source anymore. And within a few days, the outbreak subsided. So these techniques that John Snow used, which entail finding cases of infectious disease and identifying connections between sick people, these would go on to form the foundation of what we now call epidemiology, which is broadly defined as the study of the incidence, distribution, and control of disease. I want to highlight something here about the power of epidemiology. So John Snow was able to effectively stop the cholera epidemic on Broad Street, but he had no idea what caused cholera or how to treat the disease. So epidemiology works in the absence of an understanding of the mechanism of disease. Now, we're going to jump forward about 150 years to the modern era. We know that cholera is caused by a pathogenic bacterium called Vibrio cholerae, and the bacterium is shown here. For the rest of my talk, I'm going to represent Vibrio cholerae with this little cartoon. And this is really a fascinating organism for a lot of reasons. First and foremost, Vibrio cholerae is widespread in the environment. It's all over the place in salt water and in estuaries, basically any sort of marine environment. But the vast majority of Vibrio cholerae strains are not pathogenic to humans. In fact, the way one of these strains can become pathogenic and cause disease is to become infected with a virus. So as you heard earlier, there are subclasses of viruses that can specifically infect bacteria. These are called bacteriophages. And in order to become pathogenic, Vibrio cholerae must be infected with this virus. So how does this work? So Vibrio cholerae produces a protein structure on the surface of its cell called TCP. And TCP has actually been shown to help Vibrio cholerae cause disease on its own. It seems to maybe help Vibrio cholerae stick to cells within the intestine, but it's very unclear how this works. Although what's fascinating about TCP is that it serves as a receptor for a virus called CTX phage. So CTX phage can bind to TCP and then transport its viral genome into the bacterial cell. Now, once this genome has been transported into the cell, it's incorporated into the bacterial DNA. And now, the bacteria can produce virus genes. So, cholera is caused when humans consume food or water that's been contaminated with pathogenic strains of Vibrio cholerae, so strains that have been infected with this virus. And I want to emphasize, the virus isn't causing disease in humans. The virus helps the bacterium cause disease in humans. So once a human consumes a pathogenic strain, it can survive the acidic environment of the intestine and make its way to the, it can survive the acidic environment of the stomach and make its way to the more hospitable environment of the intestine. So once the bacteria arrives, it begins producing a protein that's actually coded for by this viral genetic material. And the protein is called cholera toxin. Now, cholera toxin is secreted into the intestine where it makes its way inside of intestinal cells and disrupts their normal function. So here you can actually see a real-life image of Vibrio cholerae in the intestine of a mammalian host. The cholerae bacterium have been engineered to produce a protein called GFP, or green fluorescent protein. So each individual green dot you see in here is a unique bacterium. Meanwhile, this intestinal sample has been stained with a series of chemicals to reveal the intestinal cells. So the intestinal cell DNA is shown in blue, and the boundary between the intestinal cell and the bacteria is shown here in red. So one thing I want to highlight here is the size discrepancy between the intestinal cells and the bacteria. So the blue here is the nucleus of an intestinal cell, and you can see it's much larger than an individual green dot. So these bacteria are much, much smaller than the eukaryotic cells. But what's happening in the context of this infection is that all of these little green bacteria are producing massive amounts of cholera toxin. And this cholera toxin is entering these intestinal cells, disrupting their function and causing them to excrete water into the intestine. And this causes massive diarrhea. And the diarrhea that's caused by cholera is utterly debilitating. In fact, if it's left untreated, it can kill a human within 4 to 5 hours. So an infected human can produce up to 20 liters of cholera stool each day, and this cholera stool contains trillions of bacteria. And not only does it contain these bacteria, but they've actually somehow been modified by the host environment to make them hyper-infectious and better able to infect other humans. So as you can imagine, dealing with 20 liters of watery stool is not exactly easy, especially if you don't have adequate sanitation infrastructure. And it's very common that this watery stool can leak into the environment and contaminate nearby water sources. Another human comes along, drinks from these water sources, and now cholera has made it to another human. This is how epidemics are perpetuated. So the good news is we have very effective treatments for cholera. It starts, and most times it actually ends, with rehydration with intravenous fluids. So as long as you can rehydrate someone, they're probably going to make it through cholera. Another way you can help is by giving them this special mixture of cholera salts, which is essentially dehydrated Gatorade. It basically helps restore the electrolyte balance that's often lost with diarrhea. It's sort of like Pedialyte that you might give to a child that has diarrhea. In very severe cases of disease, you might prescribe an antibiotic for a person. This has been shown to shorten the duration of the disease, but if you can keep a person alive, they'll eventually clear the infection on their own. And lastly, we have a couple moderately effective short-term cholera vaccines that can be protective for 2 to 3 years, but don't seem to have much protection beyond that. And so just to recap, John Snow's study of the Broad Street epidemic really kicks off the modern era of cholera research and establishes the field of epidemiology. We also have learned that pathogenic strains of Vibrio cholerae are all infected with a virus. And humans contract cholera by ingesting these pathogenic strains. And then cholera is extremely rare in the developed world and very treatable. So I think now would be a good time if we have any questions.
Just a question about, it's only the infected Vibrio cholerae that are actually infectious. Is it because of the binding mechanism to the intestinal wall, or is it because the cholera toxin is only produced by infected Vibrio cholerae?
So cholera toxin is only produced by infected Vibrio cholerae. The genetic instructions for producing the toxin are encoded within the viral genetic material. So uninfected Vibrio cholerae don't contain the instructions to produce this toxin.
Okay.
Great question. It's actually something where we studied cholera for decades without realizing that it was infected with the virus. Yeah?
I had a question about the slide you showed with the stained cells.
Yeah, sure.
Were the red ones dead, or just a different type of cell?
No, the red is actually, it's sort of the boundary, it's sort of an outline of the cells. So the red and blue represent different components of the intestinal barrier. Yes?
So, given that there is a moderately effective vaccine, has there been consideration for organizations like Partners in Health and people who go to Haiti and other countries after earthquakes or natural disasters to give that vaccine to try to prevent these outbreaks?
That's a fantastic question, and I'm going to ask you to hold it till the second section. I'm actually getting the signal that we need to move on, but I will definitely address that question. Okay, great question. I said question a lot right there. Okay, so let's catch up. Okay, so now I left off saying cholera is extremely rare in the developed world, but it actually still is a problem in much of the developing world, in places that don't have the same sorts of infrastructure, be it basic infrastructure like clean water or sanitation, or public health infrastructure like ambulances and hospitals and all of that. So we're going to jump back to 2010 to the country of Haiti, which has long been one of the poorest countries in the Western Hemisphere. In 2010, Haiti was struck by a massive earthquake centered just outside of the capital city of Port-au-Prince. And this was an utterly devastating blow to Haiti. It killed over 100,000 people and left millions more homeless. It also caused significant damage to infrastructure, which was severely lacking in Haiti to begin with. So in the wake of this earthquake, essentially millions of Haitians were living in close quarters, in places without access to sanitation and without access to clean water. And this is sort of the hot zone for a cholera infection. And the CDC actually looked at this, but issued the following statement. An outbreak of cholera is very unlikely at this time, while the current water, sanitation, and hygiene infrastructure in Haiti would facilitate transmission of cholera. It's not circulating in Haiti, so the risk of introduction to Haiti is low. Essentially what this means is CDC looked at the situation and said, yes, this is a prime spot for cholera, but we never saw any evidence of cholera in Haiti before the earthquake, and we think it's unlikely that cholera is going to make its way here. So I've highlighted a couple of these assumptions here in red because basically they were dead wrong. Within 7 months, a cholera outbreak was detected in a rural part of Haiti, which soon spread to the entire country. And in the years since then, over 700,000 Haitians have been infected with cholera, resulting in over 8,000 deaths. So the real question is here, how did the CDC get it so wrong? And how did cholera get to Haiti to begin with? And that's where we're going to use a tool called molecular epidemiology. So scientists looking at this situation had 3 hypotheses as to how cholera could all of a sudden show up in Haiti. The first was that a natural environmental strain, which was not pathogenic to begin with, may have rapidly evolved to become pathogenic in the wake of the earthquake. The second hypothesis was that a pathogenic strain of cholera from somewhere else in the world might have migrated spontaneously and made its way to Haiti to cause the epidemic. And finally, it was possible that cholera was inadvertently introduced to Haiti by human activity. So in order to test these 3 hypotheses, scientists decided to sequence the genome of the Haitian outbreak strain. And so sequencing the Haitian outbreak strain basically allows us to take the fingerprint of this bacterium. And by taking this fingerprint, we can compare it to the fingerprints of strains from other parts of the world. So for example, if the Haitian outbreak strain looks exactly like a strain from Asia, then that could suggest it migrated from Asia or was introduced from Asia. Or if it looks completely different from any sequenced cholera strain, we might have an indication that this is a newly evolved pathogenic strain of Vibrio cholerae. So one reason I think this story is fascinating from a scientific standpoint is because most of this work was done locally. So in the wake of the earthquake, several clinicians and researchers from Boston area hospitals traveled to Haiti to help with the outbreak response. They obtained clinical samples, so basically these are Haitian outbreak cholera bacteria that were obtained from the stool of cholera patients, and they shipped these strains back to MGH, Massachusetts General Hospital. They arrived on November 6th, which was about a week after the first indications of an outbreak were detected, and 2 days later they made their way to my lab, my boss's lab. Let's not get ahead of myself here. My boss's lab at Harvard Medical School and Brigham and Women's Hospital. And a very dedicated postdoctoral researcher grew these bacteria and extracted the genomic DNA. And he did it as fast as humanly possible. So basically he finished this at 4 in the morning, didn't stop until he had it. Shipped this to California, where a company began sequencing the DNA. So by 10 p.m., he got the DNA at 4 a.m. on the 9th. By 10 p.m. on the same exact day, we were actually receiving the sequencing data streaming over the internet. Amazing turnaround. 2 days later, the scientists that worked on this project had assembled the genome and had done some analysis of it and reached some preliminary conclusions about the origin of the strain. So to reach the conclusions about the origin of this strain, they constructed what's called a phylogenetic tree, shown here. And a phylogenetic tree is essentially a family tree. It's the exact same idea. Branches that are closer together are more closely related than branches that are further apart. And what was shocking about this phylogeny, or phylogenetic tree, is that the two samples sequenced from Haiti were very, very, very similar to strains sequenced from Asia. So just from this phylogeny alone, we have a pretty good indicator of how this strain might have arrived. And what's even more of a smoking gun is that these Haitian strains are very different from any strain that had ever been isolated in the Western Hemisphere. So this is the first time a strain looking like this had ever showed up in the Western Hemisphere. So from these data, we can actually come back to our three hypotheses, and we can rule out evolution. The fact that this strain looks exactly like one from Asia tells us this wasn't already present in Haiti. It didn't evolve. It's not a new pathogenic strain of cholera. Also, we can rule out migration, because if a strain were to have migrated from Asia to Haiti, we would have a characteristic trail that you can actually trace with cholera surveillance networks at different geographic locations between the two starting and endpoints. So it basically leaves us with this option here. Cholera was introduced to Haiti through inadvertent human activity. In the weeks that followed the publishing of this genome and the release of this data, it was shown that Nepalese aid workers working for the United Nations transferred from UN base camp basically in Nepal, where a cholera outbreak had recently occurred. And they seem to have inadvertently introduced cholera to Haiti through a defective sanitation system in their camp. So these people were very well intentioned. They were coming to help Haiti in the wake of the earthquake, but they inadvertently brought this pathogenic bacterium. And when this news reached the Haitian public, it was not received very well, as you can imagine. It resulted in a lot of rioting and protests, and all of this really just was so unfortunate because it really interfered with the response. You know, the outbreak is raging at this time, and instability in the country is only making it harder to respond to this. So there is some good news to come out of this story. In the wake of the Haitian cholera outbreak, there's been a movement to provide vaccination and also preventative care for any aid workers that might travel from a cholera endemic area to an area at high risk of cholera. The second movement, which addresses your question from earlier, is scientists actually banded together to call for a stockpile of cholera vaccines that could be deployed in the wake of a natural disaster and hopefully head off an outbreak before it happened. And this is something that I am actually a little affiliated with because my boss wrote op-eds basically calling for a creation of a cholera vaccine stockpile. In the aftermath of the cholera epidemic in Haiti, there were actually only about 100,000 doses of cholera vaccine in the world. And that is basically a drop in the bucket in the scheme of a 10 million person population in Haiti. So this is going to be fascinating right now because this stockpile has been deployed in areas of Nepal where cholera is endemic and which was recently struck by an earthquake. So hopefully it can head off any potential epidemic in Nepal. Finally, there have been calls for the United Nations to pay reparations to Haiti to basically facilitate rebuilding from this disaster. So to conclude today, I just want to leave you with a couple of these ideas, that there's a social context to outbreaks of infectious disease, and that even though we can treat them, curing a disease is really only the first step. The only way to truly prevent outbreaks of infectious disease is to be able to address the underlying societal factors which can bring these outbreaks out to begin with. And the best way of doing that was actually constructed in response to the cholera epidemic in Haiti. It's something that's sort of a comprehensive approach. And what I mean by a comprehensive approach is one that balances short-term measures like let's find sick people and treat them to make sure they don't die of this disease, with longer-term measures like improving access to quality infrastructure like clean drinking water or sanitation systems, or the public health infrastructure that can basically make it so even if an outbreak does occur, it can sort of be handled domestically and won't require, it can be nipped in the bud essentially. So with that, I think I'll finish up, and I'd just like to acknowledge our sponsors today as well as our organizers, and I'd be happy to take any questions that you may have either now or also in the wake of the second session. Yes?
Yeah, so if we stockpile vaccines, how effective, a couple questions, how effective could those vaccines be against multiple strains, or does it have to be multiple vaccines? Secondly, is there any way to purify water that's effective in killing the cholera bacteria? And third, since we don't have it in this country, if I walked into MGH with bad diarrhea, would anybody ever suspect cholera?
Okay, number one, the vaccines, there's preliminary indications to say that they would be effective in basically dampening the initial stages of an outbreak. And the idea is if you can catch it early, it won't be nearly as severe. One of the major transformations since 2010 to now is that we now have a better vaccine and we have more doses of it. And when I say a better vaccine, I don't mean the efficacy of it is any higher. It's still only short term. But the initial vaccine required something called a cold chain, which means that vaccine doses need to be kept refrigerated from production to administration, which is incredibly expensive. So now the second vaccine, hopefully, it has overcome that barrier. It doesn't require a cold chain. And hopefully, being administered in advance of a potential outbreak, it can reduce the severity of it. And third, yeah, you might surprise an infectious disease specialist at MGH if you walked in with cholera. There are a couple cases each year in the US. I believe there were 6 last year. But one of the things about cholera is that it is a very distinctive disease. There are not many other diarrheal diseases that produce such prolific diarrhea. There's also, yeah. No, it's, yeah.
One more quick question and then we have to move on.
Can you
Sure.
purify water?
You can purify water, and one of the most fascinating things about this is, so just simple filtration systems work, and there are actually at-home remedies that have been suggested. There's something called sari filtration, which is basically folding over a sari cloth and filtering water through that into a copper bowl, which does seem to be effective. There's actually a relationship between cholera bacteria and organisms in water that are not bacteria, that are larger, that can be caught by this filtration system. So there are a lot of efforts at every stage to purify water. But I think I need to wrap up. I'd be happy to take other questions afterwards.
Yeah, so you can take more questions on the poster session, but we have the psychiatric disease talk, which is in the other room. So if you want to go to that, why don't you head over there pretty quickly. And then, yeah, sorry about that. I know Troy has a lot to say, but
I do.
we can chat with him more later.
Thank you.